So a chart that illustrates the differential characteristics of the mucosa of various regions of the gut tube, it begins on the next slide. Certainly the most straightforward portion of the gut tube is the upper part of it that basically begins with the esophagus. The esophagus has a non-keratinized stratified squamous epithelium because it's mainly a conveyance tube. Note that the skeletal muscle in the muscularis externa undergoes a transition. At the superior end of the esophagus, near or at its pharyngeal junction, the skeletal muscle dominates the muscularis externa. Then the lower third typically is undergoing a transition where it typically consists of entirely smooth muscle, whereas the middle one-third of the esophagus probably contains a mix of both skeletal muscle and smooth muscle. Note that other than a stratified squamous epithelium, there are no other specialized cell types that characterize the mucosal surface of the esophagus. That changes when we begin to reach the stomach. Much of the stomach is thrown into an array of pits that extend into gastric glands. The pits tend to be shallower in the area of the cardiac portion of the stomach where the esophagus enters, as well as in the body and the fundus of the stomach. These pits will tend to get deeper as we move into the pylorus, which we'll see. Now there are distinct differences with regard to the mucosal cell types that line the luminal surface of the stomach. Basically, at and on the surface of the luminal epithelium are surface mucus cells, and as we move down through the gastric pits, they too are lined by mucus cells almost entirely. By the time we reach the tops or proximal portions of the gastric glands, now we begin to see first parietal cells, mucous neck cells, enteroendocrine cells, and finally chief cells, which typically populate the bases of the gastric glands. Functionally, mucus cells come in two flavors, as we'll see. There are surface mucus cells and mucous neck cells. They all are secreting different components of mucus that protect, obviously, the luminal surface from the acidic medium of the stomach contents. Chief cells have zymogen granules that basically contain pepsinogen and lipase precursor, which will be transformed once they hit the acidic medium of the stomach lumen. Parietal cells produce hydrochloric acid and intrinsic factor. Intrinsic factor, as we know, is essential for the binding and future uptake of vitamin B12 by the terminal ileum. Then there are a wide variety, depending on where we are in the stomach, of enteroendocrine cells that, as we'll see, produce a certain degree of variety of peptide-based hormones. As we've said, by the time we make the transition into the distal or pyloric portion of the stomach, the gastric pits have become progressively deeper, and the glands are somewhat shallower and more highly branched. Note that the mucosal cell types do not change. We still have the same array of mucosal surface and neck cells, parietal cells, chief cells, and enteroendocrine cells. Note that the EE cells, particularly populating the pyloric portion of the are producing mainly a high concentration of gastrin, which is regulating the hydrochloric acid output of the parietal cells. Finally, as we pointed out, all parts of the stomach fundamentally have four different types of cells lining the mucosal surface of either the gastric pit or the gastric gland. But note that we do need to be sensitive, and we'll point them out subsequently, about where the stem cells are that regenerate this mucosal epithelium. Now, when we move to regions of the small intestine, we again are going to find four different mucosal cell types dominating the luminal surface. Certainly the most prevalent of them, that's most frequently seen lining the duodenum, jejunum, ileum, as well as the large intestine, will be a columnar absorptive epithelial cell known as an enterocyte. These cells are characteristically contain extensive microvilli that dramatically increase their surface area that facilitates absorption. These columnar epithelial cells will basically line all parts of the luminal surface of the small intestine, and as we'll see, virtually the entire small intestine is thrown into finger-like projections known as villi, that basically in the small intestine we find folds, plica circularis, that basically increase the luminal surface area. And between the villi, the mucosa extends down into a series of crypts of Lieberkühn. So the columnar epithelial cells basically line all parts of the villi and the crypts. So it's going to be important for us to find other distinguishing characteristics that will enable us to decide whether a histologic section is from the duodenum, the jejunum, or the ileum, since fundamentally the mucosal structure is the same in all three regions. And there will be, for example. So in addition to the columnar absorptive enterocytes, these are interspersed and situated among goblet cells. Goblet cells are obviously secreting mucus to protect the mucosal lining of the epithelium. Certainly, there are going to be different populations of EE cells that are going to characterize the luminal epithelium depending on where we are. And finally, we're going to find some highly characteristic and distinct cells down at the base of each crypt of Lieberkühn. These will be known as Paneth cells. And basically, they contain secretory granules, mainly containing lysozyme, that helps regulate the bacterial makeup of the small intestine. Now, duodenum specifically. Duodenum is looking like all of the other parts of the small intestine, in that it consists of villi and crypts with the same array of mucosal cells. The distinguishing feature of the duodenal mucosa, however, is not the presence of the cells, but the underlying submucosal distinct Brunner's glands that are releasing an alkaline product into the lumen of the duodenum. So the Brunner's glands will only be found in the underlying submucosa of the duodenum, making it easy to recognize in histologic sections. Note that the EE cells, particularly in the duodenum, secrete large amounts of cholecystokinin in secretin, regulating the activity of the pancreatic ducts and their ability to release bicarbonate, as well as, in the terms of cholecystokinin, regulating bile release through the biliary tract. Now, when we move to the jejunum, note that, again, we still have the well-developed circular folds, the plicae, the villi, and the crypts, and certainly we find the same range of mucosal cell types as we found in the duodenum. The difference will be that the underlying submucosa of the jejunum lacks Brunner's glands. And the next point to make is the ileum, again, has the same array of villi and crypts and the same array of mucosal cells. So how do we tell the difference between the jejunum and the ileum? Well, certainly neither of them have the submucosal Brunner's glands, as we illustrated in the duodenum. The difference will be that the ileum will have prominent aggregations of lymphatic nodules situated under the epithelium in the underlying lamina propria. These lymphatic nodules are known as Peyer's patches. And overlying a Peyer's patch, which consists of B cells, T cells, and germinal centers, and so on, will be antigen-presenting cells known as M cells that are squamate cells situated, as we said, in the overlying epithelium adjacent to Peyer's patches. So we can kind of use process of elimination by evaluating the small intestine. When we evaluate a section of the duodenum, we're going to have the standard array of mucosal cell types, crypts and villi, but we're going to see the underlying Brunner's glands. The ileum, again, is going to look like the duodenum in terms of mucosal identity, but it's going to be characterized by many of the underlying Peyer's patches. So using process of elimination, if we're looking at a gut section that's the jejunum, we're going to find neither the submucosal Brunner's glands nor the Peyer's patches, and that will be a way of deciding that a histologic section is that of the jejunum. Finally, on the next slide, when we move to the large intestine no longer contains the villi, but it does still have crypts of Lieberkühn with a similar mucosal lining. But now there are many more mucus-secreting cells than there were present in the epithelium of the small intestine, and fewer columnar absorptive cells, respectively. But we still have Paneth cells situated down at the base of the crypts of Lieberkühn, as well as enteroendocrine cells. Now, let's be a little more precise and look at some histologic images involving different parts of the gut tube. As we said, the esophagus, and obviously beginning at the pharynx, the proximal part of the gut is the only part that's lined by a stratified squamous, non-keratinizing epithelium. And as we noted, there has to be an abrupt transition from that stratified squamous epithelium to the simple columnar epithelium that's going to characterize the rest of the gut tube, beginning with the stomach at the distal end of the esophagus. So The muscularis externa, we said, basically is slightly different in terms of the kind of muscle that it contains. We pointed out that it contains skeletal muscle exclusively in the upper third, which is really an extension of the skeletal muscle lining of the pharynx. Smooth muscle interstitial third, and probably a combination of both in the middle third. Histologic section shows the usually convoluted nature of the mucosal surface of the esophageal lining. The arrow points classically to the non-keratinizing stratified squamous epithelium. You could see more basophilia illustrated along the basal layers of this. And the stem cells of this epithelium are certainly down, like they are in skin cells, at the basal layer of this regenerating epithelium. Underlying that, the lamina propria may have some seromucous glands. Underlying that, we see obviously the array of muscularis externa that we documented as having different components of either skeletal or smooth muscle, and certainly so there's some blood vessels indicated at the arrowheads. Now, let's make the transition, and it's an abrupt one, as we said, to the stomach mucosa. Certainly we begin throughout the length of the stomach by seeing the formation of gastric pits. These form tubular invaginations that line the inner surface of the stomach, and the pits are virtually all lined by different forms of mucus-secreting cells. The transition between the pits and the glands is marked by a significant point called the isthmus. And we'll see in a moment, it is at the isthmus where the stem cells are located that regenerate presumably all of the epithelial cell types that line the stomach lumen. Then, extending from the isthmus are branched or components of gastric glands. And the gastric glands in the body of the stomach particularly are delivering up to 2 liters of gastric juice per day, and there are approximately 5 million glands throughout the entire length of the stomach contributing to that fluid volume. So below the mucus-secreting cells, we find an array of cell types that line the gastric glands. And beginning at the level of the isthmus, we'll see some mucous neck cells, and extending below that, we will begin to see first the oxyntic or parietal cells that are producing hydrochloric acid and intrinsic factor, and typically down near the base of the gastric glands will be the chief or the peptic cells. EE cells can be situated throughout the length of the gastric gland, and as depending on the area of the stomach that we're talking about, can produce and release a wide variety of gastric-based hormones. So histologically, let's look at a region of the stomach that's basically at or near the entrance point of the esophagus. And here's where the gastric pits tend to be relatively shallow, and the coiled and extensive gastric glands tend to be longer. The arrowhead is significant because it points right at the isthmus, the juncture between the deep end of a gastric pit and the beginning of a gastric gland. And it is at the arrowhead where we would find the stem cells that regenerate the epithelium of presumably all the cell types of the pit and the gland. So obviously the pit is lined by mucus cells, wide variety of 2 varieties of mucus-secreting cells that are found there. And then beginning below the isthmus, we find a prevalence of the highly eosinophilic, kind of looking like having a fried egg appearance. These are the parietal cells that are mainly obviously secreting hydrochloric acid into the lumen of that gastric gland. In addition, we'll have EE cells and mucous neck cells situated there as well. By the time we move down to the base of a gastric gland, the pale staining eosinophilic parietal cells have been largely replaced by the highly basophilic chief cells. And you can see how they dominate the epithelium extending down to the base of a gastric gland, but you can still see interspersed among them the pale staining fried egg appearance that's characteristic of the parietal cells. Now, we talked about the stem cells, and we made the significant point that they are located at the level of the isthmus, right at the juncture between a gastric pit and a gastric gland. And as we said, they are probably responsible for the regeneration of all of the epithelial cell types that make up the stomach lumen. And their mitotic rate can be influenced by the presence of gastrin and also by damage. And note that this is a highly dynamic epithelium where virtually all or many of the gastric epithelial cells are being renewed every 4 to 7 days. Next, let's look by comparison at the small intestine. Certainly, it's involved in the process of digestion and absorption of nutrients, and obviously it has multiple specializations to increase its surface area to aid in particularly the absorptive function. The duodenum is the proximal part. It begins at the pyloric end of the stomach, and basically distal to that is the jejunum and ilium, and we've helped distinguish histologically the difference between those three gastric regions. So certainly the chyme from the stomach that arrives in the small intestine is mixed with mucus secretions as well as exocrine products in the form of pancreatic juice and bile. Mucus production occurs in the surface epithelial cells throughout the length of the tract, but in the duodenum, as we said, the Brunner's glands enhance that alkaline production, as do the goblet cells in the mucosa lining the epithelium. So the rate of mucus secretion is certainly increased and enhanced by cholinergic stimulation or by chemical irritation or even physical irritation. Now, let's look at, again, the overall structural anatomy using a picture of the small intestine. So we know that the entire small intestine is thrown into finger-like projections, each of which is known as a villus, that's illustrated in this picture. Extending down deep into the underlying lamina propria is a crypt of Lieberkühn. The crypts of Lieberkühn obviously are narrow in their luminal diameter, very narrow, and separate one villus from the adjacent one. Note that the principal cell type lining both the villus and extending down into the crypts are the absorptive columnar enterocytes with their extensive population of surface microvilli. Interposed among the enterocytes will be goblet cells, mucus-secreting though they may be. Third, we're going to find a wide variety of enteroendocrine cells, mainly down in the crypts of Lieberkühn that are not illustrated on this picture. And fourth, the fourth cell type will be the specialized Pan-S cells, which are only found at the base of a crypt of Lieberkühn, whose secretory products are regulating the bacterial medium of the small intestine. The last cell type that certainly we again need to recognize, and we'll point out where they're found, is in addition to the four fundamental cell types that characterize the epithelial lining of the small intestine, we again have to be sensitive about the stem cells. And the stem cells are typically found at the base of each crypt of Lieberkühn, and they're thought to be interspersed among the Pan-S cells at the base of a corresponding crypt. Certainly, the lamina propria extends up into and fills basically the space of the villus. Lamina propria also extends down, obviously, below the crypts of Lieberkühn as well. And then we see the underlying muscularis mucosa that makes up the entire mucosal surface, or comprises the third element of the mucosa of the small intestine. Now, in the duodenum, as we said, the acid medium in form of chyme is basically being neutralized by the extensive alkaline mucus secretions of mainly the Brunner's glands that are situated in the submucosal area. And in addition, as we said, the duodenum receives digestive enzymes in the form of bicarbonate from the ducts of the pancreas and bile from the liver, and those secretory products are regulated by the release of secretin and by cholecystokinin from EE cells that line the crypts of Lieberkühn in the duodenum. And again, enterocytes, they have two major functions. Certainly, the enzymes that are present at their brush border participate in the final digestion process. And certainly their major functions is to absorb the digested food that's now in the form of amino acids, monosaccharides, and emulsified fats, and transport it from the lumen of the small intestine to the underlying vessels and lymphatics that lie in the lamina propria. Goblet cells, as we've alluded to, they are part of the lining epithelium that secretes mucus to help protect the luminal surface of the epithelium. Now, pictures. How do we know that this is a duodenum section? Well, certainly we can see by visualizing the extensive array of villi. One of them is shown at the level of where the curved arrow is. And extending down between the villi are the narrow lumens that constitute a crypt of Lieberkühn. But as we said, all regions of the gut have that as a characteristic feature. Note, just as a side issue, that the location of lymphatic tissue is not necessarily restricted to the ilium, although the Peyer's patches make it particularly prominent in that location. So the arrowheads point to some underlying mucosal-associated lymphatic tissue that might give you the sense that this is a section through the ileum, but the giveaway feature are the structures at the straight arrow in the underlying submucosa, and what you're visualizing down there are the coiled submucosal Brunner's glands, which are a diagnostic feature of only the wall of the duodenum. Next, let's talk a little more detail about the Pan-S cells. They are located at the base of crypts of Lieberkühn, particularly prominently in the jejunum and the ilium. They contain highly acidophilic secretory granules, which we'll see in a moment, and those granules basically are helping regulate the bacterial flora of the lumen of the duodenum by the lumen of the small intestine by secreting lysozymes and defensins or cryptins that help destroy and lyse bacteria. And then the EE cells, as we said, secrete a wide variety, depending on location, of hormones that interact with, in the case of the duodenum, the glands that are associated with it. Now, stem cells. We indicated that the stem cells, as we said, are commonly found at the base of crypts of Lieberkühn throughout the length of the small and the large intestine, typically adjacent to the Pan-S cells, and their job, as we indicated, is to generate by mitosis and subsequent differentiation virtually all of the other cell types that are in the lining epithelium. So like the epithelium of the stomach, this is a highly dynamic epithelium, such that like the stomach mucosal surface, all of the epithelial cells are probably renewing themselves almost every 5 days. Now, a higher magnification view is shown on the next picture. It's an H&E stained section of a crypt of Lieberkühn. And what you can see is much of the lining epithelium is formed by the somewhat eosinophilic staining enterocytes and the highly vacuolated goblet cells because the mucus secretions are washed out during typical histologic processing. The other feature that's illustrated here, indicated by letter A, are the Pan-S cells with the highly eosinophilic granules containing lysozyme and so on that are situated in their apical cytoplasm. And we know that the stem cells are situated at the base of a crypt of Lieberkühn near the adjacent Pan-S cells, and we can see that in the circular insert, a cell undergoing mitosis, that's part of the regeneration that occurs associated with the lining epithelium. Now, let's move down to a section through the ileum. Again, we're going to have villi and crypts, and you can see illustrations of them. Inside the individual villi in this particular case, at the arrowheads, are the central lacteals that are sites where basically fats have entered the lymphatic system. But the characteristic feature of this underlying lamina propria in the area of the white arrow are dense patches of lymphatic nodules that constitute a Peyer's patch. And even though we can't see it on this particular magnification of this micrograph, the M cells, the antigen-presenting cells, would be a squamous cell type in addition to all of the other cell types lining the luminal surface that would be present in the overlying epithelium adjacent to a Peyer's patch. Now, what changes when we get to the large intestine? Well, anatomically, the colon or large intestine is much larger in diameter and certainly shorter in length than the small intestine. And basically, it's where we're going to remove water and concentrate waste material in the form of fecal material before it moves from the beginning of the large intestine at the cecum through its entire length to the anal canal. The large intestine doesn't have a continuous array of longitudinal smooth muscle. Rather, it's characterized by 3 longitudinal bands positioned equidistant from one another that extend the length of the small intestine. These longitudinal bands of smooth muscle are each known as a tinea coli. And the large intestine is kind of folded up somewhat, creating pouching, and the pouches are known as the haustrations that basically are overlain by the tinea, giving the colon what we refer to histologically as a caterpillar appearance. And we pointed this out earlier, but we know that the mucosal lining of the colon has crypts of Lieberkühn but lacks the villi, which makes it fairly easy to recognize in histologic sections. And the other point that we made at is that lining epithelium is going to consist mainly of enterocytes and goblet cells, but the number of mucus-secreting goblet cells are much more significant than those goblet cells that we saw in the lining epithelium of the small intestine. Again, Lymphatic tissue may be found in the underlying lamina propria, and certainly the major function of the colon is to absorb fluid, concentration, and concentrate the ultimate waste material in the form of feces. Epithelia and histologically, here's a good view of the mucosa of the large intestine. Note again, all we're looking at now are crypts of Lieberkühn, and again, I think you can sense the increased presence and number of the light-staining goblet cells that are dispersed among the enterocytes. Panna cells would still be found near the base of a crypt of Lieberkühn, as would the stem cells.